Welcome back to Dr. Liu's VCE Biology. Today's topic is DNA manipulation techniques and applications, as presented in the 2025 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. The correct option is D, temperatures at which the experiment is conducted. Why is this the independent variable? In any scientific experiment, the independent variable is the factor that the researcher deliberately changes or manipulates to see how it affects something else. The cause, you are choosing different temperatures to apply to the DNA. The effect, you are measuring how the DNA structure, the dependent variable, reacts to those changes. Option A, rate of DNA denaturation, which is a dependent variable. The outcome or result you measure after changing the temperature. Option B, mass of DNA, which is a controlled variable. To keep the test fair, you should use the same amount of DNA in every sample. Option C, number of nucleotides, which is a controlled variable. You would use the same DNA sequence across trials to ensure the results aren't biased by the DNA's complexity. Emmanuel Charpentier and Jennifer A. Doudna won the Nobel Prize in Chemistry in 2020 for the discovery of CRISPR-Cas9 genetic scissors. The diagram below shows three different conditions that the scientists tested. Condition 1 acted as the control group, and conditions 2 and 3 were the experimental groups. The correct option is C, to direct the cutting of DNA. Why this is the purpose? To understand why C is the correct choice, look at how the components interact in the experimental groups. The scissors, the Cas9 protein, is the enzyme responsible for the actual cutting of the DNA. However, on its own, as seen in condition 1, it doesn't know where to cut. The guide in condition 2, the sgRNA and tracRNA work together to find the specific target DNA sequence. The simplified tool, in condition 3, the scientists fused these two RNA molecules into a single guide RNA, sgRNA. This molecule acts like a GPS, binding to the Cas9 protein and directing it to the exact location on the target DNA so the cut can be made. Evaluating the other options, A, to cut the target sequence, this is the job of the Cas9 protein, not the RNA itself. B, to create off-target editing, this is generally an unwanted side effect or error, not the intended purpose of the guide RNA. D, to repress the Cas9 protein, the guide RNA activates and positions the Cas9 protein rather than repressing, stopping it. The correct option is B, conditions 2 and 3. How to read the gel results. To determine if CRISPR-Cas9 was functional, we are looking for evidence that the target DNA was cut into smaller pieces. Condition 1, the control notice there is only one thick band at roughly 2,500 base pairs. This matches the original size of the target DNA. Because the band didn't move or split, we know the DNA was not cut. Conditions 2 and 3, in these columns, the original 2,500 BP band has disappeared, or faded, and was replaced by two smaller bands lower down the gel, around 1,700 BP and 800 BP. Since smaller DNA fragments move faster and further through the gel, these two bands prove that the scissors successfully cut the original strand into two pieces. Why the other options are incorrect? A, conditions 1, 2 and 3. Incorrect because condition 1 shows no cutting occurred. Cas9 cannot function without a guide RNA. C, condition 1 and 3. Incorrect because condition 1 is the control group where the DNA remains whole. D, condition 1 only. Incorrect, because condition 1 is the only trial where the system failed to edit the DNA. The correct option is D. CRISPR, Cas9, can be guided to edit DNA by combining sgRNA and tricRNA into a single guide RNA. Logic behind the hypothesis. A good hypothesis predicts the outcome of a specific change. In this experiment, the scientists were specifically testing if they could simplify the natural system. The before in nature, condition 2, the system uses two separate RNA molecules, sgRNA and tricRNA. The prediction, they hypothesized that if they fused these two molecules into one single guide RNA, sgRNA, the system would still work perfectly. The proof, the gel electrophoresis results for condition 3 show the exact same cutting pattern as condition 2, proving their hypothesis was correct. Why the other hypotheses fail? A. Incorrect. Depends entirely on sgRNA without modifications. This is contradicted by the fact that they purposefully modified the system to create a single guide RNA in condition 3. B. Incorrect. Cas9 can cut DNA without guide RNA. The results of condition 1, the control, proved this wrong. Without RNA, no cutting occurred. C. Incorrect. The tricRNA sequence is unnecessary. The diagram shows that the tricRNA, or its sequence in the sgRNA, is a key component for the system to function. The answer, C. Father C. The logic, an offspring, the lamb, inherits half of its DNA from its mother and half from its father. To solve this, you must match the bands, exclude the mother. First, look at the lamb's bands and find the ones that match the mother. Match the remaining bands. Any band in the lamb that doesn't come from the mother must come from the biological father. Why Father C is correct? If you look at the lamb's bands that are not present in the mother, specifically the second, fourth, and sixth bands from the top, Father C is the only candidate who possesses all of those specific matching bands. Why others are incorrect? Fathers A, B, and D all lack at least one critical band that the lamb possesses, but did not inherit from its mother. The answer, B, the charge of the DNA molecule. The logic, gel electrophoresis works because DNA molecules have a consistent negative charge due to their phosphate backbone. When an electric current is applied, the negatively charged DNA is repelled by the negative electrode and pulled toward the positive electrode. Why the other options are incorrect? A. The ribose sugar, DNA contains deoxyribose, not ribose, which is found in RNA. Regardless, the sugar does not provide the motive force. C. The number of DNA fragments. This affects how many bands you see, but it doesn't assist the physical movement through the gel. D. Types of nitrogenous bases. The sequence of bases A, T, C, G does not change the charge or the speed of movement, only the length of the fragment matters for speed. The correct option is D, repeating the cycle many times. Why this is the correct choice? PCR is often described as a biological photocopier. The word amplification in the question refers to creating millions of copies of a specific DNA segment. The mechanism, a single PCR cycle consists of denaturation, annealing, and extension. However, one cycle only doubles the DNA. The goal, to get enough DNA for testing, like the gel electrophoresis seen in your other images, the scientists must repeat the cycle, usually 25 to 35 times, to achieve exponential growth. A. Binding of RNA polymerase. PCR uses DNA polymerase, specifically Taq polymerase, to build DNA, not RNA polymerase. B. Heating to 72 degrees Celsius during annealing. 72 degrees Celsius is typically the temperature for the extension stage. The annealing stage usually happens at a lower temperature, around 50 to 65 degrees Celsius. C. Attachment of primers during denaturation. Primers attach during the annealing stage. Denaturation happens at high heat, 94 to 98 degrees Celsius, to separate the DNA strands. Splicing. A single continuous strand containing only the coding sequences, exon 1, exon 2, and exon 3 joined together. The non-coding introns must be removed. 5' methyl G cap. A protective cap added to the 5' end of the molecule. 3' poly A tail. A chain of multiple ADE9 nucleotides added to the 3' end. Recombinant DNA technologies are used to produce large quantities of human insulin to treat diabetes. Shown below are some of the steps involved in one method used in the production of human insulin. 2 steps are incomplete. Complete step 4 and step 8 in the spaces provided below. Step 4 DNA ligase. The answer: DNA ligase is added to join the insulin genes, chains A and B, into the plasmid vectors by forming phosphodiester bonds. The logic: while restriction enzymes act as the scissors to cut the DNA, DNA ligase acts as the glue that permanently seals the gene of interest into the plasmid, creating a recombinant plasmid. Step 8 Amino acid chains A and B. The answer: amino acid chains A and B are bonded together, typically via disulfide bridges, to form the functional, mature insulin hormone. The logic: in this specific method, the 2 chains are produced separately by different batches of bacteria. For the insulin to actually work in a patient, these 2 separate protein chains must be chemically joined to reach its final quaternary structure. The enzymes, the tools, restriction endonucleases. Acting as molecular scissors, these enzymes cut DNA at specific recognition sites. DNA ligase, known as molecular glue, it joins DNA fragments by catalyzing the formation of phosphodiester bonds. DNA polymerase, used to build new DNA strands by adding nucleotides, crucial for the extension stage of PCR. 2. Key techniques: PCR, polymerase chain reaction, a process used to amplify DNA through repeated cycles of denaturation, annealing, and extension. Gel electrophoresis, a method to separate DNA fragments based on size, driven by the negative charge of the DNA molecule. CRISPR-Cas9, a powerful gene editing tool derived from bacteria that uses a guide RNA, sgRNA, to direct the Cas9 protein to cut specific DNA sequences. 3. Practical applications: DNA profiling, utilizing gel electrophoresis to identify individuals or determine paternity, like the lamb example, by comparing unique fragment patterns. Recombinant DNA and transformation, inserting human genes like insulin into bacterial plasmids to mass-produce essential hormones. GMOs and TGOs, the creation of genetically modified organisms and transgenic organisms to improve crop yields or nutritional value. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.